Carbohydrates show up everywhere on the MCAT, but can you quickly tell the difference between a monosaccharide, a disaccharide, and a polysaccharide under pressure? In this crash guide, we're stripping carbs down to their essentials: structure, classification, and the key reactions the test loves to ask about. By the end, you'll not only recognize these molecules instantly, but actually understand how they behave so you can answer faster and with confidence on test day. Carbohydrates are polyhydroxyaldehydes or ketones that serve as key energy sources and structural molecules. At their core, carbohydrates are organic molecules made of carbon, hydrogen, and oxygen, typically in a ratio close to 1 to 2 to 1, which is why their general formula is often written as CnH2n-on. The defining feature is that they contain multiple hydroxyl groups and either an aldehyde or a ketone functional group. If the carbonyl is at the end, it is an aldose. If it is in the middle, it is a ketose. A simple way to remember this is A comes before K, so aldose has the carbonyl at carbon 1, while ketose shifts inward, often to carbon 2. On the MCAT, you may be asked to identify whether a sugar is an aldose or ketose based on its structure. Beyond structure, carbohydrates play crucial biological roles. Glucose is the primary energy source for cells, especially in tissues like the brain and red blood cells. Carbohydrates also form structural components, such as cellulose in plants and chitin in exoskeletons. These structural roles often depend on differences in glycosidic linkages, which we will discuss later. Additionally, carbohydrates are involved in cell recognition and signaling when attached to proteins or lipids as glycoproteins and glycolipids. On test day, expect passages that tie carbohydrate structure to function, such as how glucose can be rapidly mobilized for energy, or how structural polysaccharides resist digestion. Understanding both the functional groups and biological roles gives you a foundation for tackling more complex carbohydrate questions. Monosaccharides are classified by carbon number and functional group, with stereochemistry playing a critical role in their biological function. Monosaccharides are the simplest carbohydrates and cannot be hydrolyzed into smaller sugars. They are categorized first by the number of carbons, such as trioses with 3 carbons, pentoses with 5, and hexoses with 6. Glucose, for example, is an aldohexose, meaning it has 6 carbons and an aldehyde group. Fructose, on the other hand, is a ketohexose. Another key classification is stereochemistry, which is extremely high yield for the MCAT. Most monosaccharides have multiple chiral centers, leading to D and L configurations. These are determined by the orientation of the hydroxyl group on the chiral carbon farthest from the carbonyl. If the hydroxyl is on the right in a Fisher projection, it is D. If on the left, it is L. A helpful mnemonic is D is right. Importantly, human metabolism primarily uses D sugars. Monosaccharides can also form cyclic structures in solution, creating hemiacetals or hemikettles. This introduces a new chiral center called the anomeric carbon, leading to alpha and beta anomers. Alpha means the hydroxyl is trans to the CH2OH group, and beta means it is cis. On the MCAT, you are often asked to identify D versus L, alpha versus beta, or recognize how many chiral centers a molecule has. Mastering these classification systems helps you quickly interpret carbohydrate structures in both discrete and passage-based questions. Disaccharides and polysaccharides are formed through glycosidic bonds, whose type determines digestibility and function. When 2 monosaccharides join, they form a disaccharide through a condensation reaction, releasing water and forming a glycosidic linkage. These bonds are named based on the carbons involved and the orientation, such as alpha 1 to 4 or beta 1 to 4. For example, Maltose consists of two glucose units linked by an alpha 1-4 bond, while lactose has a beta 1-4 linkage between glucose and galactose. A key MCAT concept is that humans can break alpha linkages but not beta 1-4 linkages, which is why we cannot digest cellulose. This is a classic test question. Why can humans digest starch but not cellulose? The answer lies entirely in the stereochemistry of the glycosidic bonds. As we move to polysaccharides, structure becomes even more important. Starch and glycogen are storage polysaccharides, with alpha linkages that allow for branching, making them more accessible for enzymatic breakdown. Glycogen is especially highly branched, allowing rapid release of glucose during energy demand. In contrast, cellulose is a structural polysaccharide with beta 1-4 linkages, forming rigid, linear fibers. On the MCAT, you may be asked to compare these molecules or predict properties based on bond type. Always focus on linkage type and branching, as these directly determine biological function and digestibility. Carbohydrates undergo key reactions such as oxidation, reduction, and esterification that are commonly tested on the MCAT. One of the most important reactions is oxidation. Aldoses can be oxidized to form aldonic acids, and stronger oxidation can convert the terminal carbons into dicarboxylic acids. A classic test for reducing sugars involves their ability to be oxidized, such as in Benedict's or Tollens tests. Reducing sugars have a free anomeric carbon that can open into the linear form and act as a reducing agent. On the flip side, reduction reactions convert the carbonyl group into an alcohol, forming sugar alcohols like sorbitol. These compounds are often used as artificial sweeteners and can appear in passage-based questions. Another important reaction is esterification, particularly phosphorylation. Glucose can be phosphorylated to form glucose 6-phosphate, which is a key step in glycolysis. This connects carbohydrate chemistry directly to metabolism, a major MCAT focus. Additionally, glycosidic bond formation itself is a type of condensation reaction. Recognizing these reaction types helps you interpret experimental setups and metabolic pathways. On test day, if you see a question about whether a sugar is reducing or non-reducing, focus on whether the anomeric carbon is free. If it is involved in a glycosidic bond, it cannot act as a reducing sugar. These reaction patterns are predictable and highly testable. Carbohydrates are central to metabolism, especially in pathways like glycolysis, glycogenesis, and glycogenolysis. Glucose sits at the heart of energy metabolism. In glycolysis, glucose is broken down into pyruvate, producing ATP and NADH. This pathway occurs in the cytosol and does not require oxygen, making it essential under anaerobic conditions. Glycolysis is one of the most frequently tested pathways on the MCAT, so understanding how glucose enters and is processed is critical. Glycogenesis is the process of storing excess glucose as glycogen, primarily in the liver and muscles. This occurs when energy is abundant, such as after a meal. In contrast, glycogenolysis is the breakdown of glycogen to release glucose when energy is needed, such as during fasting or exercise. These processes are hormonally regulated, with insulin promoting storage and glucagon promoting breakdown. A helpful way to remember this is that insulin puts glucose in storage, while glucagon gets glucose gone into the bloodstream. Carbohydrates also connect to other metabolic pathways, such as the pentose phosphate pathway, which generates NADPH and ribose 5-phosphate for nucleotide synthesis. On the MCAT, you may encounter passage questions linking carbohydrate metabolism to energy balance, hormonal control, or disease states like diabetes. Understanding these pathways allows you to integrate biochemical knowledge with physiology, which is exactly what the exam is designed to test.